process of discovering, testing, and eventually gaining approval for selling a drug is a long and arduous one. Let's have a look at the traditional pipeline for a pharmaceutical small molecule. You want to start from a research finding and bring it all the way up to a patient. But before you're allowed to treat patients, you will need a so-called marketing authorization. The marketing authorization is given by regulatory authorities. In Europe, this is the EMA, the European Medicines Agency. Once you obtain marketing authorization, you are permitted to treat patients. In order for the regulatory authorities to grant you marketing authorization, you need to provide them with evidence that the drug that you have developed is safe and efficacious. Efficacy meaning that the drug works. You submit the data demonstrating safety and efficacy to the regulatory authorities. With EMA, it is called Marketing Authorization Application, or MAA. As you see, we have been starting quite at the end of the development process. This is because in the MAA you summarize the evidence that you have collected so far in the clinical development part and in the preclinical development. We will look at all the different stages of a typical development process now. The research and development process typically starts with target identification. A target is a molecule in the body that is intrinsically associated with a particular disease process, and you expect that this molecule in the body could be modulated by a drug, and this modulation will produce the desired effect. Once you are confident that your identified molecule is indeed a good target, the milestone you reach is called target selection. Once you selected your target, you're looking to find a small molecule that can modulate the target. To identify those molecules, you typically use a process called high-throughput screening. With high-throughput screening, you initially screen hundreds of thousands of molecules. Those molecules that interact with your target are called hits. These molecules are then further optimized with respect to properties such as solubility and toxicology, resulting in so-called lead compounds. During lead optimization, the lead compounds are further optimized, and the molecule with the best properties is selected and nominated as a candidate drug. Candidate nomination is a very important milestone as the research phase ends and the development phase starts. The goal of preclinical development is to prepare the safe administration of the candidate drug to humans and to collect evidence for efficacy. For this, model systems such as animals, in silico models, tissues, or organoids are used. Toxicology studies are used to demonstrate that the candidate drug is sufficiently safe to administer to humans. The information collected through this phase is then used to request approval to conduct a first-in-human clinical trial, the FIH application. Once you obtain approval, you can start with the clinical development. The clinical trials are organized in several phases with a growing number of participants. Phase 1 is the clinical trial phase where you assess the initial safety, dosing, and how the drug behaves in the human body. This phase is often done in healthy volunteers and not yet in patients. Nowadays, phase 1 is sometimes preceded by a phase that is called phase 0, where you do microdosing. In phase 2, you typically move to patients, and during this phase you are looking into efficacy of the drug, in other words, indicators that the drug improves the disease. In this phase, you might further optimize the dose and keep on monitoring for side effects. Phase 3 trials are similar to phase 2, but on a larger scale, both in terms of numbers of participants and duration. In addition, they usually compare the candidate drug to other available treatments. If you consider your trials to demonstrate efficacy and safety, you can file for marketing authorization with regulatory authorities. The regulatory agencies will review the submitted information, and if they decide that it's efficacious and safe, you will receive marketing authorization. However, the process actually doesn't end here. Once a medicine reaches the market after clinical trials, its safety and possible adverse effects are continuously monitored, because there could be, for example, rare side effects that were not seen in clinical trials. The process of monitoring and evaluating the safety of medicines is called post-marketing surveillance or pharmacovigilance.